Hello everyone, my name is Khushal Ingle, and today we will discuss about the term that is genetic code, or specifically we will learn here about the codons. So as we know that the central dogma of molecular biology is nothing but it is about the all the process which is nothing but formation of protein from the DNA. So here you can observe that from the DNA the formation of RNA takes place. That RNA may be of rRNA, that is ribosomal RNA, messenger RNA, or the tRNA. And the process of formation of RNA from the DNA is called as the transcription. Now further, this RNA undergoes the translation and will form the final product that is called as the protein. So nothing but all this is it is about the central dogma of molecular biology. So here we have to learn about the codons. So when we learn about the codon, these three different terminologies we have to consider, that is anticodon, Wobble hypothesis, and codon. But in today's lecture, we have to discuss about only about the codon. So starting with the first point, that George Gamow in 1954, he first of all coined the term that is genetic code. And after this, the genetic code was discovered by Nirenberg and Matthey in 1961. Apart from this, Marshall Nirenberg, Severo Okuya, Hargobind Khorana, and Francis Crick and Matthey deciphered about this genetic code. DNA and RNA contains information which is written in a chemical language of nitrogenous bases such as adenine, guanine, cytosine, and thymine. In case of the RNA, that thymine may be replaced by the or it is that thymine is replaced by the uracil. So in case of the DNA, you will observe there is adenine, guanine, cytosine, and thymine. In case of the RNA, there is adenine, guanine, cytosine, and the uracil. So this coded information of the DNA or the RNA should be transferred in nothing but protein. So the cell require a method to translate this language into the protein, and it uses this genetic code. So with the help of this triplet code, nothing but it is the genetic code. So with the help of this triplet code, the four bases can supply 64 different combinations. These combinations are nothing but these are sufficient to code for 20 different amino acids. You can observe here that this is the chart in which the possible 64 codons are given by the Hargobind Khorana and the other members. So these uracil, cytosine, adenine, and the guanine. So these letters which are present in the what first letter means on the fifth prime. Then this is the second position of this letter, that is uracil, cytosine, adenine, and the guanine. So this one is the second letter, and this third one is came from this third letter, that is uracil, cytosine, adenine, and guanine. So if you combine this first, second, and third, you will get these different 64 codons. So on this basis, the scientists or the researchers they made this table where you can observe the different codons. So all these different codons will code for different amino acid. So you can observe here that, for example, this one, this first will code for the phenylalanine, and second one will also code for the same, that is phenylalanine. Then another one, this will code for the leucine, and this one is also for the leucine. So these six combination will code for the leucine amino acid. In this way, the different code will code for the different amino acid. Unit of each three letter, that is three nitrogenous bases of an mRNA, is called as the codon. The nitrogenous base which makes codon are selected from either adenine, guanine, cytosine, and uracil. As we have seen in previous table, there were three possible conditions were there. So out of these four nitrogenous base, any three may be present within the codon, and that codon will code for the different amino acid. This coded information is transferred to mRNA during the process, that is transcription. So I already explained about these nitrogenous bases which are present in DNA or the RNA. Now very important point about the codon, that is characteristics of the codon. So one by one we will learn about the different characteristics which are shown by the codon. So first one is that genetic code is universal. So this chart or the table that we have learned in previous slide, that is about the codons. So this genetic code is universal. So each and every organism you will observe the same codes. Yes, there are some exception cases are there which will code for the different amino acids, but universally this code is the same. Second point is that genetic code is commaless. It means that it do not follow, it do not contain any space when that mRNA will read by the ribosomes. At that time it will continuously read. So this genetic code is commaless, it is continuous. Then genetic code is degenerate. It means that more than one codon can be used for a specific amino acid. So if you remember the previous table in which we have learned that more than one codon can be used for specific amino acid. So if you see in that table specifically leucine, that leucine was coded by the different codons. It means that more than one codon was used for the formation of the leucine. Then, and this property is called as the degenerative property or the degenerate. Then genetic code is non-overlapping. One letter can't used for two different codons. The next one is that genetic code is non-ambiguous. It means that a specific codon will always code for same amino acid. So here in this table, if you see randomly, for example, AUG, so AUG will always code for methionine, and above that AUG, AUA is there, which will always code for the isoleucine, and below the AUG, there is GUU that will code always for the valine. It means that genetic code is non-ambiguous. So a specific codon in this table will always code for the same amino acid or a specific amino acid. Now about the initiation codon. So initiation codon, it is also called as the start codon, which will initiate the protein synthesis. Here the first codon will always the AUG. So AUG, this codon will always code for the methionine. So it is the universal start codon. In some cases, for example, bacteria, it may code for the formyl methionine. Then in this way, the number of codons will be present in mRNA and that will code for the different amino acid. Then next one, that is stop codon or nonsense codon. So this codon do not code for any amino acid. That's why the process of formation of the protein, or that is the translation, will stop here at this codon. That's why it is also called as the stop codon. These stop codon do not code for any amino acid and they will always present at the 3' of the mRNA or at the last position. And there are three types of the stop codons, that is UAA, UAG and UGA. So already we have seen in that table that there were these three codons which were not coding for any amino acid. Next is that the codon has polarity. It is read from 5' to 3'. So it always having a polarity. So for example, if I take this AUG, that this one will be the 5' and this one will be the 3'. So we will always read from 5' to 3'. Here also again UAA. So we will read this codon in this direction, that is 5' to 3' direction and never from 3' to 5' direction. So it was all about the codons. These are my references which I have used for this video. So I hope that you will be able to understand the concept behind this codon. Thank you so much.